Hello, everybody. Welcome to today's lecture. In the previous lecture, we have talked about the lower level of motor system, which including the muscle properties and muscle force generation. And we also see different kinds of reflex loop in the spinal level. So today, we are going to look at a more general aspect of the human behavior. And we'll go through the spinal cord upwards. So we'll look at the higher level control of the motor system. So at first, we will look at go through several experimental cases that have demonstrating particular features of movement kinematics. In the first case, let's start with this case. This is about the planning of timing and with respect to the target accuracy. So this shows the relationship between your movement velocity and the requirement of accuracy. And intuitively, we would imagine if we move fast, we should be less accurate. While we will go slowly, we could be accurate at target. And this experimental study will demonstrate it in the more detail about this aspect. So here we ask a participant grasp a pointer and on a table, for example, and he can start from different initial positions. And the task is to reach to a target line with different speed. So the movement with different move times. So movement times in this experiment is controlled. So it can be 140 milliseconds, 170 or 200 milliseconds. And during the experiment, the experimenter can control the movement times if the subject moves too fast or too slow. For example, they set like 10% threshold if the subject is not finish the movement in the required desired duration, the movement will discard it and repeat it. So they vary 3 initial positions and with 3 different movement times that are required. So in total, we have 9 conditions. And here exactly we plot 9 dots here. And we plot in the axis is the average speed. So for each condition, the participant will repeat several times. And on the y-axis, it is the variability of the target position. So the standard deviation is calculated with respect to the mean. So it shows that how variable is your end position is. And what we see here, so this shows that once you have larger speed, you become more variable, or in other words, you are less accurate in the end position. That also shows that since you have higher speed, you also have higher force. That's also related to higher force will also lead to more variable. And but in this case, the target line is fixed. But what about if we can vary this target region? So we still have this A. This is the distance. We can have different distance. And here this W is the width of the target. So you can the experimenter can change different weights such that the subjects can reach inside the target. And here it's plots. So there are several curves, and each curve is represent one kind of target. So with one certain width of the target. So it could be narrow or wide, and we have different distance, which is A. And subjects are instructed to move as fast as possible. So here we have introduced this index of difficulty, which is the logarithm of twice the distance divided by the target width. That means, so if you have very far targets, but you have very far movement distance, but you have very narrow target, that is more difficult, right? And vice versa. So if we plot this line, so in the logarithm, and we see that there's a linear relationship between the index of difficulty and the movement duration. And for A and B, it can be fitted by the regression of this model. And this this relationship, this equation is usually referred to the Fitts law. So here it's more like cognitive, the planning of the timing of the movement and how accurate you arrive at the target. And let's see another case. The second case about still about the velocity of the movement, but this is with respect to the curvature. So curvature, as you know, this is the one divided by the radius. For example, if the subjects were required to move like this figure of 8, like 2 circles, one big circle and one smaller circle. For the larger circle, so they have a larger radius. That means they have smaller curvature and vice versa. So subjects can do this kind of periodic movement. And what is plotted here? So they plot velocity for each period, and in the same time they plot this radius. And we see if the velocity is larger at the larger radius. So like, for example, here, the radius is larger, so the velocity is also larger. And at the lower radius, they have lower velocity. And the same case for the other condition, which is the one circle is inside the other. So here we can plot, we don't use radius, but we use these curvatures, one divided by radius. And here y-axis is the velocity. And we see, by the way, this velocity is the tangential velocity, which is measured in meter per second or centimeter per second. And here we show, this is actually, we can also do the regression of this curve. And we see this actually, this relationship is an exponential function here. And this is usually we refer to the power law. And this is, we can see another case. So instead of the periodic movement, we can also ask the participant to write a letter. For example, this letter A. So they have to start from from position 1 and like this at 5, they stop, and then they do at 6 and stop. So what we see is actually a similar results. So they plot this axis is the movement time. So and then we plot the velocity, also the tangential velocity. At a different position, you see, for example, the position 2 and 4, they have a larger radius, which means they have smaller curvature here at 2 and 4. And then in this case, the velocity is also larger. Velocity larger at smaller curvature, right? This is also consistent with the previous results. And what is also can be noted is that we see the point 5 and 6. Like these 2 points, movement direction is inverted. And we see 5 and 6, actually, the velocity is almost 0. That is also makes sense. Sorry? Okay, there's no question, right? Okay. So we see the velocity is almost 0 at 5 and 6. And that means this at these 2 positions, they have a very small radius. That means the curvature is kind of infinite, and that's why you have 0 velocity according to this power law. Okay, so I hope this is clear. And also, okay, so now we have a look at more. This is the kind of cognitive aspect of movement control, which means you have to like find the relationship between the planning of timing and movement speed, also the accuracy. But if we can also look at individual trials, so which means that we can look at the time course of the trajectory of the movement itself. And let's see what we can find here. And this is a very classical study in human motor control area. And this is this setup is also very standard. This is called a manipulandum. Normally, this is a robot, but here it's just like a handle with 2 joints, flexible joints. And the participant is grabbing the joint and it moves on a horizontal surface. So the movement is only restricted to two joints, which is the shoulder joint alpha and the elbow joint theta here. And the subjects can move between different targets. There are in total 6 targets. So this basically is just a reaching movement, like point-to-point reaching movement. And we all know that we always produce straight line. So usually we move like in straight. But there are some like special cases. For example, if you move vertically, and then you will see it's not the shape usually is not very straight, and also particularly when you avoid obstacle, you cannot really move straight. But these are only special cases. But usually, if there's no external restrictions, people always tend to make straight trajectories in the hand, right? And what is interesting here, so if you plot the hand speed, the velocity of the hand, you see no matter which target, which direction you are moving, so you always have this symmetrical peak. It's called a bell-shaped hand velocity profile. So it always preserved this profile across different conditions. And if we look at joint angles, we see in different conditions, we will have different combinations of joint angles. And these patterns in joint angles seem to be more complex, but hand is kind of hand trajectory and velocity, hand velocity seems to be invariant. And what does it mean? So it seems that if we are planning a movement, it seems that our brain is planning in the hand. It's more much easier for us to plan a hand trajectory rather than joint angles, right? If you consider our brain control individual angles, that means they have to find this all the nonlinear combination of this joint angle. It becomes more complicated. So planning in the hand velocity is much easier in this case. Okay, and here is another example showing this velocity profile. So in previous condition, we see these different movement directions, but here we have the same movement direction, but we varied the distance of the movement. And we see the velocity also, although they have different velocity, but always there's this bell-shaped velocity profile. And even if we normalized across different times, actually they almost overlap for all the conditions. So this is another demonstration of the hand velocity profile, and this is important. So this leads to a theoretical study of the question, how can we produce this hand bell-shaped hand velocity? So here we introduce a variable called jerk. Jerk is the third derivative of position, or the derivative of acceleration. And people found, Hogan, 1984, found that if we define this jerk variable, and we can have an algorithm that's trying to minimum the jerk across the movement time, like the square of the jerk. And we define this as a cost function. If the algorithm is to minimize this cost, we can have a solution that produces smooth position and a bell-shaped velocity profile. So this model can generate smooth movement and the experimental observed hand velocity. So this is a simulation study. If you are more interested in the mathematical details, you could refer to this web page. It has a more detailed mathematical derivation of this algorithm. So let's look at again about another case. So this is more complex. Here we still have this setup, as you remember, so this robot manipulandum. But here in the robot, they have these two joints. They put two torque motors that they can program the torque motor such that they can apply a certain force at the handle. So once you remove the handle, it can be, you can feel some disturbance on the hand. So if there's no disturbance, so you just turn off all the motors, the subject's it's called a null field. That means there's no external force. The subject is moved straight line to to the 8 targets with straight line. That's not surprising. And then in the next condition, because they can program the 2 torques to apply different force on the handle, and this is called a velocity dependent force. I will explain what is force look like. So you see here we have hand velocity in x direction and in y direction. So velocity dependent force means that the faster you move, the stronger perturbation or stronger disturbance you will have. And for example, here, if you stay at 0 velocity in both directions in the middle of this plot, there will be no perturbation. And if you, for example, if you move in this direction, so you move along the y direction, and then according to this profile, you will get a horizontal perturbation on the hand. So it means if you move straight on the y-axis, your hand will be deviated to the side, right? And the same if you move the horizontally, for example, you move along x direction, you will have a vertical perturbation of the hand. And this kind of this is also very typical perturbation that used in many human movement studies, particularly in the hand reaching movement. And so as a result, when you move and you encounter these perturbations in the hand, you see the subjects cannot, in many cases, they cannot reach the target anymore. And here, for example, if you move like vertically, then you will have a horizontal perturbation, and then you will have deviation, so you become not accurate in reaching the target. However, we can ask the subject, because when the subjects learn, they can give like a certain, if they don't change this force characteristic, so subjects can practice many trials. And because they know, like, they can gradually learn this force field. So what we see that after several trials, the subjects can learn to adapt to this force perturbation such that they can still arrive these targets accurately. And interestingly, they move this straight line. So after adaptation, you see, we compare the null field, the kinematics becomes very similar as the null field. But this is only happens after learning our adaptation. And in experiment, when this force perturbation, so after adaptation, when the experimenter suddenly turned off the perturbation force, so the subjects didn't know that, but they just turned off. So we see this called after effect. So the subject is actually moving in the opposite direction as the force that would apply. And this is called after effects. So what is important here is that we see that both null field and adapted case, subjects always tend to make straight movement. Or maybe this is could be easy to interpret because you always want to make simple, like you don't want to generate much cost. So with the straight line, it's much easier. So you could interpret it like this. So till now, we have talked about mainly the behavior measurement in the human participants, and clearly that our brain is controlling this movement, because we see each time the planning, the trajectory, and velocity and so on are all different. It must require certain kind of brain control. And as in last lecture, clearly the muscle and spinal cord are not able to deal with these complex external conditions. So we need to understand the brain. So let's try to look at the brain. So from now on, I will talk about the brain structure. Before we come to human, I would like to show a comparison between human brains and animal brains. And here we see the animal brains from the simple animals like fish and amphibian and so on. And here we have this human brain. And what we notice that the human brain is definitely larger and more complex in structure than the animal brain. Notably, we see this human brain have these folders like with these curved gaps. This is usually called sarcos. And so that means because our scalp has a certain volume, and with this folded structure, you can have more area in the cortex, in the surface of the brain. So once you stretch of this folded area, you will see a very large area of the brain. So this is more efficient to store the space, consider the space. And here, this is another plot if we compare cross species. So we plot the body mass and the brain mass. And in one plot, we see for different species, for some species, they have very low level. So we consider this slope. If we consider slope, some species like sheep or pig, they have very low ratio of brain to body mass ratio. While for other species, particularly in human, you see if we calculate the slope, human has the largest ratio of the brain to body mass ratio. Also, the octopus or the chimpanzee have also a larger ratio, and the dolphins seem kind of outlier. But it also have a relatively lower speed than human. So okay, so this is just like some kind of descriptions of this physics of the brain structure. So now let's come to the human brain. So I will only talk about the brain area that directly related to movement. We have like brain is very complex structure, and they have many areas that are not directly related to the movement generation. So in this lecture, I will talk three brain structures is considered the most important for movement generation, which are motor cortex, cerebellum, and basal ganglia. So in the following, we will look at them one by one. But before that, I would like to give you a brief overview of the brain structure. Here is just like the 2D representation of the brain. So here I actually bring a real physical model of the brain, as you can see here. So the brain have two hemispheres, left and right. So here is the side view of the brain like this. And this view is like you cut like from the top of the brain, but it's not very easy to look at inside. So here I will first go through several structures, but maybe we are not learning all of them. But I will just point you there are some important structures. So here's motor cortex, but motor cortex usually refer to including the premotor side of cortex. So some people call premotor cortex also motor cortex. So the terminology a little bit different across different researchers. And we also talk cerebellum. Cerebellum is here, like here. If you see, it's here, or in this figure you can see it's here. Okay, and also the basal ganglia. Basal ganglia is this two light brain area. So this structure is this three structure we are going to talk. But there are also other structures. For example, this thalamus is also important. This is considered as a relay of all the sensory signals. So once you have the spinal cord, because you have sensory signals going up through the through the spinal cord to the brain, and almost all these sensory signals has to come first, arrive at thalamus, and then will be processed in the thalamus and then go to other areas of the brain. So thalamus is an important relay of the sensory information. We also have the brainstem. structure is roughly here, but it consists of also several substructures. So we'll not talk about the brainstem, but this is one of the old structures in the brain. Okay, so let's look at this model, and I will take it off so we can see this side of the brain. So we'll talk about the motor cortex. It's about here, and then here we have the cerebellum, and the basal ganglia is roughly at this position. So I will first start with the motor cortex. So here we show the connection of the motor cortex with the lower level, the spinal cord. And there's a direct connection from the neurons, which is located in motor cortex, and they send axons to synapse in the spinal cord. And this is called cortical spinal tract. So this is the most direct way for motor cortex to control the spinal cord muscles or the spinal cord motor neurons. And the descending pathway, also there are many other descending pathways through the red nucleus or reticular and so on, but this is, we are not talking about today. And anatomically, we can see these different descending pathways will arrive at different locations of the spinal cord. And here we have, if we still remember from the previous lecture, we have these motor neurons here in the lower part. So what is interesting here, we see this inhibitory interneurons. So why we would have these interneurons? So in humans, there are actually many direct pathways. So like this, so from the brain directly synapse on the motor neurons. But in animals, particularly in lower level animals like rats, they only have this descending pathway. They have to always go through the inhibitory interneurons, and they don't have actually these direct connections. But the direct connection gradually increase when the species comes higher. For example, in monkeys, they have relatively more direct pathways, direct connections from motor cortex to motor neurons. And in humans, they have the most amount of these kind of connections. So the motor cortex can consist of three parts. So the primary motor cortex, it's usually we refer to M1, and premotor area, PMA, and supplementary motor area. For the SMA, we are not going to talk about this. So we mainly focus on M1, PMA today. So here we see where they are located, this PMA and M1. So M1 is located just beside the central sulcus. And also here, this S1 is the sensory motor cortex, sensory cortex. This receives all the sensory information from the spinal cord. But we are not going to these structures. So let's start with M1. If we slide, make a slide from the vertical direction, and we cut here, and we look the side of this M1 structure. And what we found, so these neurons have connections to the muscles, and actually we could map this location, different locations or different spot to find where this spot linked to which muscles. How did we find this connection? And that is a very old study by a Canadian neurophysiologist called Penfield. And he's a doctor, and when he do operations in patients, when they open the skull, so they can see the brain, and then he put the electrodes, like they can send some electro stimulus, and they put electrodes at different locations of the M1 area of that patient. And once he gave a stimulus and a certain location, he can observe a certain muscle activation. For example, if we put somewhere here, and then he will see a twitch in the face muscle. So he know, okay, this point is linked to the face muscle, and this point is linked to the arm muscle. And in this way, we use this micro stimulation and observe the muscle response. He mapped this mapping and put different body segments in the different spot of the M1. And this is usually called homunculus. It's because it's kind of a figure of the human body, which is mapped to the M1. What is interesting here is if we look at the distribution of different body segments, we find the hand area. So from the thumb to the little finger actually occupied almost the most, has the most portion of the cortex area, also the hand, wrist. So it seems like a very large portion of M1 area contribute to the control of hand movement. That is also not surprising because our humans are very good at manipulation tasks with the fingers if we compare to other animals. Okay, and then so this is the basic anatomy of this M1. Let's look at what the neurons in M1 do. So this is an experimental study in monkeys. This is an invasive study because they implant electrical board in the monkey's M1 area. So this device is kind of electro array that can have many electrodes, and they implant in the brain so they can record the activity of the surrounding neurons. But to identify which signal is which neuron, they need complex mechanisms or algorithms to reconstruct the activities. But we just have to know that with this technique, with implanting electro arrays, they are able to recording neurons in the M1 area. And in this experiment, monkeys were asked to make reaching movement. So the monkey is grasping a joystick. They can move different target in different angles, 0, 90, 180, and 270. Actually, there are 8 directions. And let's take an individual neuron in M1 areas. I will explain this figure here. So we see here we have 8, the monkey reaches in 8 different directions. So each with 45-degree interval. So this is one single neuron. And here each vertical bar represents one action potential of the neuron. And along this axis is the timing. So if you see, okay, this bars is very intense here, that means that this neuron is more activated. And here each line represents one trial of the same neuron. And here, for example, this represents the monkeys does 5 trials in 90 degrees reaching movement. And this area is the 0 is the movement onset. And we see in this direction, 90 degrees, at around the movement onset, this neuron in the M1 area becomes more active compared at the movement onset. And also this neuron is also active at other directions, for example, in 135 and 180 and 225. But this neuron is not active or even inhibited in other directions. If the monkey is moving, for example, in 0 degree direction or 315-degree direction, we see there's almost no activity in these neurons, in this neuron. And then we can plot a curve. So x-axis is the movement direction, and y-axis is the how strong is this neural activity. So we see for this neuron, we have at 135 degrees, they have the maximum activation of this neuron, while in the other direction, the activation is less. And it seems that this individual neuron is mostly tuned, or is tuned mostly to be active at this particular direction. However, if we use only this neuron to control direction, it will be very less accurate, because they are active at basically from like 90 to 200 degrees. They are all active. So we cannot rely on this single neurons. And then we introduce a population of neurons. So if we recording, say, hundreds neurons, because each neuron has this kind of property. For example, this neuron is more active in this direction, but the other neurons may active in the other direction. So at each movement direction, so we have we can plot this tuning curve for individual neurons, and we take each movement direction, we can get the activity of that individual neuron, and we plot it together. And this is this shows the population activity of these neurons. And when at certain direction we sum up all the neurons, we see the sum of this activation is pointing to the movement direction. Although it's not perfectly aligned, but we see clearly, like if we rely on the group of neurons, it gives us more accurate prediction of the movement direction. So so this study showed that single neurons give you not a very accurate estimation of the movement direction, but rely on the population coding, we can have a better estimation of the movement direction. So that's this kind of property of this neural population in M1 area. So we consider they do a direction coding function. And this is important because it leads to the next slide. So we introduced the concept of neural trajectory. And this trajectory is directly calculated from the population vectors. So this time we calculate the population vectors in the time course. So if this is still a monkey experiment, so if we ask a monkey to do like a movement in the time course, and at each individual time point, we can reconstruct this using this population vector, we can get the direction tuning at this particular point. And then we can calculate the next point. Then we can have the direction, how this neural trajectory is the kind of the direction of this velocity. So in this case, we can reconstruct the trajectory of the population of these neurons. And this case is a finger movement. And we see if we plot this trajectory, this is the kinematics of the finger movement. But if we reconstruct this neural trajectory based on population vectors, we see the neural trajectory is actually very similar, like the finger trajectory. And this kind of higher level representation of the kinematics of the movement. And here, as we have talked about this kinematic regularity, we talked about the power law. So here we still take these figures from the previous slide, but we plot this again to show the relationship between the curvature and the velocity. But note this time the plot is angular velocity, it's not tangential velocity, but it's basically the same principle, but just mathematically it's different. So if we plot angular velocity versus curvature, then this is just an example. It's not related with this experiment. So we see this so-called two-third power law. So in this case, it becomes a linear relationship between the angular velocity and the curvature. And this, if we use this neural data, we can also reconstruct a similar this two-third power law in the level of brain neurons, population of neurons. So that shows this lower level kinematics regularity is also presented in the higher level neural activities. Okay, so let's continue with, so this is about M1 neurons. So what are the neurons in premotor areas doing? So here we still have monkey. Unfortunately, we have to always use monkeys because in humans it's very difficult to get access to the neural activities in the brain. So okay, so this poor monkey is asked to do another task. So they have to respond to a signal. So they have to wait. First, have to wait in front of the screen. And once there's a instruction, the light is on, the subject is going to press a button. So what is recorded is a neuron in the premotor area in this monkey. We see the actual potential is very small in when the monkey is doing nothing. It's kind of a resting activity. But once the stimulus is on, so the light is on, but the monkey is not moving yet, but he knows that, okay, I have to move now. And then we see this neuron in premotor area becomes active. So this is before the moving, but the monkey knows that he's going to move. But once the monkey starts to move, so you see this, once the monkey produces a movement, this neuron becomes silent again. So this means this neuron is in premotor area is only active after the monkey has the intention to move and before the monkey actually executes the movement. So that is the property of the neuron in premotor area. And there's another type of neuron, very interesting neuron called the mirror neuron. It's also located in the premotor area. So again, this monkey to ask to do different tasks. So the task is to pick up a peanut. The monkey can pick up the peanut by himself. And then once the monkey is doing the movement, this neuron is active, becomes active. And in the second case, when the monkey observes another monkey do the same movement to pick up this peanut, but the monkey itself doesn't do anything. And we see the same neuron also fires in this case. And if we use a human to pick up the peanut, the same movement, but with a human, and the monkey is still watching but not moving, you still see this activity in these neurons. And in the other case, once the experimental doing another movement, so they're not grasping with hand, but using a tool to pick up the peanut. So this is a different movement as the previous one. And in this case, when the monkey is watching, this neuron is not firing. And that is why it's called a mirror neuron. That means it fires both when the monkey is doing the movement itself, and when the monkey is watching the other monkey or human is doing the same task. Okay, so this is motor neurons, mirror neurons. Sorry. Okay, so I think we have talked about the motor cortex. Now let's come to the next part of our brain, this cerebellum. Cerebellum is also an important motor structure, which is about the coordination of the movement. Here we have an overview of the connections of cerebellum with the other areas of brain. So cerebellum is here. Or maybe I can first show you. So here, so we can see maybe on the right side, we see the structure of cerebellum is also like two hemispheres. And so this is the like 3D structure of cerebellum, but here you only see the 2D picture. So okay, let's come back again to these connections with other brain areas. So cerebellum is connected with this is part of the thalamus. It's called the ventrolateral thalamus. And it's also connected with the pons, which is from the brainstem. So it doesn't directly connect with the cortex, but there's the information flow from the cortex through the pons and to the cerebellum. And this picture, we see more closely how everything is connected. I want to mention this red nucleus. I think we see this part before in previous slide. This is also very important structure in movement coordination. But we are not going to the details of red nucleus. But here we have to know that the cerebellum, the output of the cerebellum is connected to the red nucleus. So this is one output here. And the other output of cerebellum is connected to the primary motor cortex and premotor cortex, although it has to go through the thalamus to make this connection. And once it reaches primary motor cortex, then it goes down to the spinal cord to control the muscles. And also for the red nucleus, also the cerebellum control also has to go through the red nucleus and then go down to the spinal cord and control the muscle. So from here, from this picture, we see there's no direct connection from cerebellum to the spinal cord. It always either to go through red nucleus or go through the motor cortex. But however, actually recently, just several months ago, I was at a conference, a motor control conference, and a researcher has reported a study that she actually found a direct cerebellum connection to the spinal cord, but it's in rats, it's not in humans. And she demonstrated that actually there's also a connection. There could be a connection, at least in rats. So the cerebellum can directly send signal to the spinal cord. I also want to briefly explain how this researcher found this connection. Also about other researchers, for example, these connections, how they discover these connections. Again, actually, many of studies are done actually in monkeys because we consider our brain is very similar like in monkeys. So in monkeys, there is technique using virus. So they inject the virus, a certain particular type of virus in a particular area of the brain, of the monkey's brain. And the virus, after the injection, the virus will spread. So they will propagate along the neurons to go to other neurons. And this, the delay of this propagation can be predicted. For example, we know, like for example, in 1 week, the virus should cross 1 synapse. So they should go to 1 neuron to the next level of neurons. And in 2 weeks, they have to go to 3 or the 3rd neurons. So they know the relative timing for the neuron to cross different segments of the connections. And in experiment, they could look at using some technique, they can trace the spread of the neuron, and then according to okay, how far is the neuron is, then according to this timing, they can calculate how far the neuron travels in certain time, and then they can look at for example, in one day the neuron goes from cerebellum to the red nucleus, and they know, okay, there should be a direct connection from cerebellum to red nucleus. Otherwise, there will take 1 week if there are several connections in between. So this is virus injection as a technique like to probe this circuitry, but unfortunately still in monkeys, not in humans. Okay, so let's continue to look at more details of the cerebellum structure. So it is the cerebellum consisted of several parts, but here we actually only look at the surface of the cerebellum, or the cerebellar cortex. So this is the surface, the structure. And here we see we have 3 layers. So this lowest layer is called granular layer. That is the input layer to the cerebellum. So in the granular layer, we see there's a very thin fiber called mossy fiber. It carries the sensory or afferent signals into the cerebellar cortex. And the mossy fiber will synapse with also these very tiny cells called granular cell. They synapse here in the granular layer, and this is considered as one major input or the afferent input to the cerebellum. And the second layer is called Purkinje cell layer. So this layer is considered the output of the cerebellum. So in this layer, there's this relatively large cell called Purkinje cell. This cell send signals as output from the cerebellum. And here, the third layer is called the molecular layer. And we see the molecular layer, there's many these axons from the granular cell, this kind of a parallel structure. It's called the parallel fibers, which are the axons of the granular cell. And this layer, because they synapse also with the dendrites of the Purkinje cells, it's considered to be an information processing layer. So they process the information at this layer. Also, another input to the Purkinje cell. So we said there's a mossy fiber as the input, but there's also another input from the climbing fibers, also to the Purkinje cell. So there are basically two afferent input to the Purkinje cell, and the Purkinje cell sends the output as the cerebellum output command. So this is the basic anatomy of cerebellum. Of course, there are also other small cells like Golgi cell and basket cell and stellate cell. So we are not going to these details. Okay, and this is about anatomy. So here we have from the control engineering perspective, Wolpert has proposed a control scheme which is related with the cerebellum structure. So here, let's start talk with this so-called inverse model. So what is the inverse model? That is, for example, if you want to produce a movement, so you want to, for example, I want to make a straight line, you have to know which command my brain has to send to produce this movement. So here, inverse model, the input is the desired movement, and the output is the command of your brain. So this what is the inverse model does. So here this is a control scheme. So let's go to first this feedback loop. So assume that your command is a desired trajectory, and through a feedback loop, so you have actual trajectory, and this feedback controller operates this trajectory arrow and give you a modified motor control, motor command to the plant, to the controlled plant. But in the meantime, the arrow also goes to inverse model. So this arrow helps to update this inverse model because usually you have the link that you know, if you know the desired trajectory, you know what control command you should produce by this inverse model. But if the situation has changed, if the environment has changed, you then which result in movement errors. So you have to update this inverse model. So you have to know, okay, in this, if I still have this desired trajectory, but I have another condition, which movement command I should produce. So you have to update this inverse model through the learning of these errors. This is the control engineering scheme. And in this study, they related this inverse, or they propose that the inverse model is actually located in the cerebellar cortex. Why? As we see in previous structures, if you still remember the cerebellar cortex, first they have these mossy fibers as the control input. So mossy fibers basically carry all the sensory input to the cerebellar cortex. So here they consider this is this actual trajectory is actually the sensory input that goes to the cerebellar cortex. And then next, this multifiber will synapse with the granule cells. And the granule cell will connect with Purkinje cells through this parallel fiber. And then the Purkinje cell will send the final output from the cerebellar cortex to other areas. So, and also here, this from this way, from the climbing fiber, they assume that the climbing fiber represents the error of the feedback. So the feedback command, and they can update this feedback inside the cerebellar cortex. So basically, this is an anatomical map to this control scheme, but the exact implementation in neural level is still is not solved. Maybe here there's the Purkinje cell also have a simple spike and complex spikes, but we are not going to the more details of these properties. So this model is linked with cerebellum, but here Volpert also proposed other kind of model. It's called a forward model. So forward model is the opposite of inverse model. So forward model means that you have a motor command, and this command goes to the forward model, and this model can simulate what the output should be. So inverse model is just the opposite. The input is what is the desired trajectory, and the output is the command that you want. And this forward model is usually used for prediction of a movement, because we know that the sensory feedback is usually have very large delay, at least 20 milliseconds. So the nervous system, particularly in the brain level, they may not rely on the sensory feedback because of this delay. And in this case, how do you still make an accurate movement, or how do you still can estimate to reach the target? So for this, you have to predict using to do prediction using this forward model. And you can combine forward model and inverse model together. Here they show a parallel structure with multiple pairs of forward and inverse models. So they can be used for controlling different aspects of the task, or even for different tasks. But this is more details and more complex. So if you want to know more about this control engineering scheme, you can go to this Volpert paper. This is a review paper which covers all these aspects. Okay, so there's another more detailed model. So this is a simulation for reaching movement, but I will not go into detail of it. But we see that it also takes account many brain structures. So, and they assume that certain variables are controlled through the, for example, in motor cortex, they have this acceleration as input and torque as output, and so on. So this is another simulation studies, which is more concrete implementation in mathematics. But we'll not look at this in detail. So this is cerebellum. Next, and also another aspect is about disease. Actually, the understanding of cerebellar function has been advanced by studying patients with cerebellar diseases. So here, let's see what happens in people with the deficits in cerebellar function. So what we see in these patients is mainly a problem in coordination and the timing. In the first example, this patient has a left cerebellar lesion, so which means the left cerebellum is damaged. And unlike the motor cortex, so cortex you always cross, so left cortex control the right side of your body, but in cerebellum, so the left cerebellum is controlling the left side, the same side of the muscles. And then what you observed at the same side of the lesion, on the left side, your left hand, when you respond, you do a movement in response to a go signal, the left hand has problem with the timing. So there's a delay in the lesion side. And also about coordination. This is a typical clinical examination when they check the patients. So this is the so-called finger to nose test. So you ask the patient to move your finger to the nose. So normally as healthy subjects, there's no problem. You just make a straight line to your nose. But in cerebellum patients, they can still reach the nose, but you see the curve, the trajectory is becomes very complex, and there are also some kind of oscillation. They have some problem in generating more coordinated behavior. Also, when we ask the patients to do rhythmic movement, so like you flip your hand, and normal subjects, you do this periodic movement, but in cerebellar patients, they are not able to generate this rhythmic movement. And this is another example showing the coordination of the joints. In this example, we have a control subject here, control participant, which is healthy, and a participant with cerebellar damage. So the task is the same. They ask the participant to flex the elbow joint while you keep your shoulder joint stable. So you have no difficulty doing this. So you move your elbow joint, but your shoulder joint can be stable. But in cerebellar patients, they are not able to keep their shoulder joint stable. They always move together with the elbow joint. And let's look at the more details about the torque profiles in both participants. What we found here. So if we look, we plot different torques at the joints. So the muscle torque, net torque, and interaction torques. And what we see here, if you look at a muscle torque and interaction torque, which is the red and light green line. So in the control participant, the muscle torque is very accurately compensate the interaction torque, which is the light green. So you see they are almost at the opposite side. But in cerebellum patients, she generates the muscle torque is not able to compensate. They cannot the opposite shape of the interaction torque, and that's why they cannot stabilize the shoulder joint. So this is more like in the torque level of this study of this disease. Okay, next we will look at the next very important, and this is also, I think this is a very, very interesting structure. It's called basal ganglia. So what does basal ganglia do? So this is basal ganglia is very important for voluntary movement. So you have the intention you want to move. So usually we take it for granted. So once you decide to move, okay, I will just send a command out. But that is not the case. So usually we don't feel that there's a problem in the healthy conditions. But if we look at the patients, we will see that later. The patients with basal ganglia deficits, they are not able to initiate. So once they have the intention, they cannot move. So you can go or not go. They cannot balance. So they hesitate. They cannot really produce intended movement. So it's kind of a modulation. It's balance between go and not go. So we will see in detail later. So how it looks like. But in general, it's a kind of a modulation of movement generation. So let's start with the basic structure of basal ganglia. So we see basal ganglia is connected directly with the cortex, particularly the motor cortex. And it also sends feedback to the motor cortex. And this is through the thalamus. This is called the ventrolateral thalamus. So it's through this. So this is kind of a feedback loop, which means if your motor cortex, because motor cortex directly controls spinal cord. So once the motor cortex initiate a movement, they are not just sending to the spinal cord, but they are also sending to the basal ganglia. And the basal ganglia do some operation of the signal and feedback to the motor cortex. So normally this modulation is no problem with healthy people. But if there are deficits, and this feedback modulation will lead to very strange movement, which we will see later in later slides. So let's still start with the anatomy of basal ganglia. So here we see where the basal ganglia is located in the brain. So we don't have to remember or to know these names, but we have to know that there are 5 parts of the basal ganglia, which is showed here in this figure, so where they are located. So I can also show you in the brain. So if we look at from inside, the basal ganglia is roughly at this location. It's quite in the center of the brain. So, okay, so let's know that there are 5 components of the basal ganglia, because later in the later we will put these 5 structures as 5 blocks, and we will see the connection between these 5 blocks. So in this slide, exactly we have these 5 block striatum. So it's 1 block, 2nd block, substantial nigra and globus pallidus, 3rd block, and the nucleus subthalamus, the 4th block, and the ventrolateral thalamus, the 5th block. Although this is thalamus, part of thalamus, but it's always considered within the basal ganglia structure because it's very tightly connected. Okay, so we have 5 blocks shown here, and we have the motor cortex, which give command to the basal ganglia, and they receive feedback from basal ganglia. So this is a loop, closed loop. We have to know that there are neurotransmitters. So what is neurotransmitters? These are chemical messengers that act between 2 neurons, can be either excitatory or inhibitory. But whether it should be excitatory or inhibitory depends on the type of neurotransmitters. For example, glutamate is an excitatory transmitter. That means if the previous neuron sends out glutamate, and then the next neuron will be excited by glutamate. And if it's GABA, then it's sending GABA. That means the neuron will inhibit the next neuron, which is connected. And there are a special neurotransmitter called dopamine. And dopamine can be either excitatory or inhibitory, depending on the receptor of the dopamine. So here we have 2 types of receptors, D1 and D2. So D1 is an excitatory receptor that will cause excitation once they receive dopamine, and D2 will cause inhibition when they receive dopamine. In this scheme, I also have to explain that. So here, this like the diamond shape means inhibition, and this arrow means excitation. These 3 colors. This green color represents glutamate, which is the excitatory neurotransmitter. And the red is GABA, which is an inhibitory neurotransmitter. And the dopamine, depending on the receptor type, so they have 2 types, the orange and the yellow one. So this orange goes to the D2, which is inhibitory, and the D1 is excitatory. Okay, and then we see, again, these 5 blocks. And we see the substantia nigra has 2 parts, and here globus pallidus also has 2 parts. And now let's try to understand this loop. First, let's assume that we take out of the nucleus subthalamus. So assuming that this part doesn't exist. And then everything comes from striatum is GABA, right? So this red inhibition. So everything is striatum will send GABA neurotransmitter, And then again, through these two blocks, they also send GABA continuously, and then this is excitatory. So that means you have a cortex is send glutamate, which is excitatory, and they use twice GABA, like twice negative and one positive, which means, so if this doesn't exist, that means from cortex to basal ganglia and back to the cortex is one positive, two negative, one positive, which means positive. That means this loop is excitatory. So once cortex excites basal ganglia will excite cortex. So this is called the direct loop. And there's an indirect loop. Now let's put this one in, this guy, nucleus subthalamus in. So here it has this guy produce this glutamate which is excitatory, and then this leads to additional pathway. So here, for example, if you come to the GABA, twice GABA here, and then it forms another road from here, it excite to back to the globus pallidus, and then again GABA, and then again excitatory. So for example, this loop, you have inhibitory, excitatory, inhibitory. So 3 times inhibitory, and which means from cortex, if it goes to nucleus subthalamus, this path will be inhibitory. And similarly, if we go this way, to the left, it also 3 times inhibition, right? GABA inhibition, and then this is also inhibitory. So which means here we have one direct pathway which is excitatory, and two indirect pathway which are both inhibitory. We will look at these two pathways in the next slide. Here I also want to mention that part of the left part, this part of substantia nigra is producing, the task of this structure is producing dopamine. And if this part has problem, it will cause a disease called Parkinson's disease. We will see it later. And this is important to know about this. Okay, so let's continue with the direct and indirect pathway in the next slide. So here is basically just a replot or reorganization of these different structures. So here's the direct pathway from cortex and back to the cortex, which is plus minus minus plus, which is excitatory. And we have this indirect pathway. So through the subthalamic nucleus, which is, if we just count the minus plus, if we go through the indirect pathway, it will be inhibitory. And normally, so here you see in this yellow area, this is termed the indirect pathway, and this part is direct pathway. And usually, if you like to look at these two arrows here and here, this orange arrow, usually these two direct and indirect pathway are balanced. That means the excitation. So once you have a input from cortex to basal ganglia, it produces both excitation and inhibition back to the cerebral cortex. And this excitation and inhibition are usually balanced. And we see here the two arrows are at the same width. But if there's some problem in this structure, you will see these two arrows will be unbalanced, and this will cause disease. First disease is called Parkinson's disease. So what is Parkinson's disease? Usually this disease is caused by the loss of dopamines, because they, if you remember, I mentioned that the substantia nigra is producing dopamine, and in these patients, these cells are degenerated and they are not able to produce dopamine anymore. And what we see from the behavior, the Parkinson patients are always freezing. If they want to move, they are freezed, they cannot move, and they also have tremors. but we don't know why these neurons that produce dopamines die in these patients. And let's see what happens in Parkinson's disease. So as we said, we have the D1 and D2 as the receptors of dopamine. Once this guy produces less dopamine, so it has effect on both excitatory and inhibitory D1 and D2 pathways. But since D1 and D2 are not the same, the sensitivity is not the same to this decrease. So this will cause unbalanced. For example, this D2 is more sensitive to the decrease of dopamine. So this arrow, this inhibition will become stronger. And this arrow, which is the direct pathway through D1, becomes weaker. And this will cause imbalance in the final output. You see, if you just count, you can count like this plus and minus. And this, because of this imbalance, it will result in a final result will be less excitation in the cortex. This means in Parkinson's patients, once he wants to move, they generate a moving command in the cortex. And this command going through the damaged basal ganglia and will inhibit back, will send the inhibition back to the cortex to inhibit this command. And that's why we see these patients, once they want to move, they cannot move because it's this inhibition caused by the degeneration of dopamine neurons. Okay, I hope it's better explained with Parkinson's disease here with this scheme. And another disease which kind of the opposite effect of Parkinson is called Huntington's disease. Huntington's disease is very complex, and I only talk one of the symptoms of this disease is called chorea. It means the dancing. It's probably like a dance. They have involuntary jerk movement. And of course, Huntington's disease has other problems. For example, in the early stage, they have a loss of memory and so on. But we only focus on related to this movement problem, this chorea symptom. But we see they do more involuntary movement, but this movement are still coordinated. That means coordination means like the cerebellum and spinal cord are not damaged, so they can still produce coordinated movement. And the cause is, I think it's still largely unknown, but many people think they are because of the genes. And let's look at what causes this dance in Huntington's disease. So as I said, so in Parkinson, there's problem with this guy, but in Huntington's disease, they have problem with the connection from the strium or the caudate putamen. So this is one structure of the basal ganglia. So they have the problem from this structure to this connection to globus pallidus. So this pathway is degenerated in this patient in Huntington's disease. And because this degeneration, it will cause imbalance of this direct and indirect pathways. And still, we can count the plus and the minus, and we will see the result of this loop is the excitation to the motor cortex, which means if the Huntington's disease have some very small intention to move, or even they don't have intention, so some random excitation from the cerebral cortex, and through this this positive feedback loop, it will amplify this activity back to the cerebral cortex. And that may explain why this Huntington's disease have this involuntary movement. So now I want to show you a video about Parkinson's disease, and we will finish soon after this video. So I will try to first quit this mode. I'll show you a video about, can you see this YouTube video? I hope so. If not, please let me know. Okay, So this is a Parkinson, a patient with Parkinson disease. We see there's a large tremor in the hand, and the patient is not able to move. So he wants to move, and he can walk very slowly, but almost his movement is inhibited. Okay, if the experimental helped him a little, he can move a little bit. And this is not stable. And finally, he falls down. Okay, and the second video is patient is doing riding a bicycle. And we see he has virtually no problem with riding a bicycle. So he can do cycling, and he will later even able to make a turn with the bicycle. And maybe I will have let you to think about the question why it could be like this. So maybe one possibility is that you see once this patient he didn't start the bicycle, the experimental has to push him to start. And once he start, okay, let's see here. You see once he stopped, he cannot move again. So he will freeze at the same place. Okay, so let's come back to the slides. Oh, sorry. Let's come back to slides. So these patients, once he voluntarily initiate movement, he is freezed. But once the experimental helps to push him and he can do this cycling, it seems that the cycling movement is more like reflex-based. Because if you think, if this movement is done by the lower level, it's not involving basal ganglia, then everything's fine. But once he wants to do a voluntary movement, you have to go through this loop, go back to the cerebellar cortex. So then it's always inhibited. So that means this cycling movement may be involved less with this cerebral level. So it's maybe more contributed by the spinal level control or cerebellum control. And the similar thing about walking. So the Parkinson's patient sometimes can also walk, but with reduced intention. So he should not think that I'm doing this movement. So let everything comes automatically with the lower level control. So with very less consciousness, not voluntary to walk. And then it seems this kind of locomotion may require lower level reflex based modulation. And we will see how locomotion is controlled in the next lecture, where Rashid will talk about control of locomotion based on the reflex loop. So for today's lecture, I would like to summarize some key points. So we first look at the kinematic regularity. We start with the fifth law, which considers the velocity and target accuracy trade-off, and we talk about the power law when the subject is doing this scratch or drawing a circle. And we also see the invariant of movement kinematics about the bell-shaped velocity profile, which can be produced by the minimum jerk model. We also see the force field adaptation task, where subjects restore the straight line movement after adapt to a environmental force. And the second part, we talked about the brain level control of movement, and we talked about the motor cortex, and we talked about cerebellum, which is coordinates movement, and the basal ganglia is kind of modulation, excitation, inhibition, and this balance, and keeps that ensures the smoothness of movement production. Okay, we will finish here, I think.